So, in chapter 14, we talked about Mendelian inheritance, Mendelian patterns of inheritance, and we talked about two laws that Mendel put forward, the law of segregation of alleles and the law of independent assortment of alleles. So Mendel put forward that there was no blending, right? When genes, we now know from mom and genes from dad, come together in baby, when baby makes gametes, those genes separate once again. And we also talked about the law of independent assortment, right? When baby who has genes from mom and dad goes on to make gametes, right? Just because the gamete gets the gene for mom's nose doesn't mean the gamete will also get the gene for mom's ears, right? It might get dad's ears and mom's nose or vice versa. It's totally independent. These traits independently assort. They assort 100% independently, or do they? Turns out they don't. This is why I always say that there are exceptions. Some genes, it turns out, are linked. And it was Thomas Hunt Morgan, again, who figured this out. So he was studying his flies again, and he looked at two different traits. He looked at fly color. So most flies were gray. That was the wild type, but there was a mutant group that had an allele that made them black. And wing length. So most flies had long wings, which means they could fly. There was a mutant allele that gave flies vestigial or short wings, wings that were not functional. And if you cross these guys, so you end up getting, a F1 generation, which is all dominant. So in this case, that means gray and long-winged. But if you then do a test cross, where you cross that individual, again, with someone who's homozygous recessive for both body color and wing length, then you should get 50-50, right, for each trait, or 25% for each combination of traits. So essentially, 25% should have long wings and be gray, 25% should be long wings and black, 25% should be short wings and black, 25% should be short wings and gray. But that is not what he found. He found that if you were gray, you almost always had long wings. If you were black, you almost always had short wings. So instead of 1 to 1 to 1 to 1, right, equal ratios, he found almost 1 to 1 to 0 to 0. Obviously not 0 to 0, but those traits, gray color and long wings, tended to travel together, right? The parental chromosomes tended to stick together. And this was new, right? This indicates that those two traits, body color and wing length, did not assort independently. Well, why wouldn't they assort independently? Right? Turns out they are on the same chromosome, right? So as you might expect, traits that are on the same chromosome, that come from genes on the same chromosome, even if the chromosomes assort independently, as they do, those traits will not independently assort. But they're not always linked. There were individuals that had, for instance, gray body and short wings. Well, that's because you get some level of crossing over. And crossing over can split up those two traits, even if they're on the same chromosome. So genetic recombination allows a shuffling of those traits between mom and dad's chromosomes. All right. So the parental types are the types that have, black and short and gray and long together. Any of them that had black and long or gray and short were recombinants, right? You had traits that came from mom and dad together on the same chromosome, and that indicated that there had been some crossing over recombination that had occurred. It turns out that how frequently you get recombinant chromosomes rather than just parental chromosomes can tell you about how close two genes are to one another on the same chromosome. So if genes are on two different chromosomes, they will travel together about 50% of the time, which means they're sorting independently, right? Right. If I get my mom's nose goes into one gamete, there's a 50% chance that I'll get it together with mom's ears, and a 50% chance that it'll go with dad's ears, right? So that's separate chromosomes. If they're on the same chromosome, it could be less than 50%, right? And the closer they are on a chromosome, the less likely they are to separate, right? The less often you get recombinant chromosomes, and the more often you get those parental chromosomes. The further away they are, the more likely that a recombination event was to have happened in between there. So in this case, parental type offspring and recombinant offspring, looking at wild type and black and vestigial and normal wings, right? Out of 2,300 total offspring, there were 391 recombinants, and so about 17% of the time you get a recombinant. That is much less than 50% of the time. In this case, that percentage dictates how far apart they are. So 17% we call 17 map units apart. It turns out this was discovered in, surprise, surprise, Morgan's lab again, but it was one of his graduate students named Alfred Sturtevant, and he predicted that the farther apart two genes are on a chromosome, the more often recombination will occur, and he used that to make genetic maps, or sometimes they're called linkage maps, because they're based on how often or how linked two genes are. And again, each percentage of recombination frequency is considered a map unit. So if two traits are on the same chromosome and they only get taken apart from one another because of recombination 1% of the time, right? Remember our body color and our wing length, right? Those should have only traveled together 50% of the time if they were independently assorting, but it was 17% of the time, meaning they're on the same chromosome. We would consider that 17 map units apart, right? If they were only separated 1% of the time, right? We'd call that 1 map unit apart. This doesn't give exactly the location on the chromosome, but it does give the relative location on a chromosome. And if you are to compare maybe 3 different traits, then you can start to get a pretty good map going. So we know that this gene and this gene, right, B and VG, so body and vestigial, they cross over 17% of the time, right? They recombine 17% of the time. We know that body color and then this other gene cross over 9% of the time. Wing size and this gene cross over 9.5% of the time. So this is probably somewhere in the middle of that. And again, these are all estimates. Right? Now imagine if you did this with lots and lots and lots of genes. Right? You can come up with a pretty good estimate, pretty good relative map of where these genes are on a chromosome compared to one another. And if you combine that with some staining, some chromosome banding, you can get a pretty good map, right? What we would call a cytogenic map of where things are on a particular chromosome.